Hello, everyone. Thank you for this opportunity to present my final project on my deep learning course. So I'd like to start by saying that the topic of my final project is the category of drug discovery. So let's get right in. The motivation for this is that currently response to cancer therapies is widely variable. We have data available in humans and animal models, but animal models do not translate well into humans. The available human data is in the form of clinical trials. They're expensive, sparse, and extremely hard to extract information in structured manners, just piles and piles of unstructured text. Cell line models of cancers, which we create and interpret in our group at the Broad Institute, are a useful proxy to perform high throughput screens of drug responses. So this is scalable to thousands of samples across multiple cancer types. The data is structured and uniformly measured. But some of the drawbacks are that cell lines are homogeneous, whereas tumors are not. And cell culture and passaging in labs introduce experimental artifacts that are unique to cell lines and that cannot be translated to tumors. And overall, this is just drug response experimentation in a screened manner, in a pooled manner, is just extremely noisy. So many thanks to the DepMap, the Broad Institute, this is where I work, and Sanger Wellcome Institute databases. So here's a problem statement. Given molecular characterizations of cell lines, can we use deep learning methods to model the response of each cell line to cancer drugs? So for the input, and I'm going to go over these in detail in a bit. So we're going to be given the mutation profile. We have the gene expression available. We have gene essentiality and tissue type. And the output or dependent variable is a drug response for each of 495 drugs, which is the IC50. So a little more expansion into these input modalities. What is a cancer cell line? A cancer cell line is just a mini cancer on a dish type of model. They're in vitro models. They're derived from patient tumors grown in the lab. Gene expression means a cell line by gene matrix of normalized gene expression values. These capture the transcriptional state of a particular of a set of genes in each cell line. Mutations are represented as just binary matrix that tells you if a mutation is present or not in a cell line. CRISPR gene effect, this measures gene essentiality based on impact on cell survival. So in a pooled manner, each gene is knocked out using CRISPR, and then the cell viability, as in the cell survival rates, are measured as a response to that perturbation of knocking that gene down. So if a gene is essential to a cancer, that means knocking that gene down will make the cells die quicker than a gene that is not particularly essential to that cancer. And then the last one is lineage, which is just the tissue of origin of the cancer cell line. It could be pancreas, it could be brain, it could be your lymph cells, or so many other, or uterus, or so many other different tissues. A little explanation on the response variable. So IC50 is a drug dose that reduces viability to 50%. Cells are treated with increasing drug concentrations, and viability is measured at each dose, and it's fitted to a curve that is shown here. It's just an illustration. And IC50 is inferred from the fitted curve. So lower IC50 indicates higher drug potency. Moving on to the technology and the model that was used. So this is implemented on KRAS. Autoencoders were used to encode gene expression and CRISPR data into latent features. Lineage and mutation data were converted to embeddings, and inputs were combined into a two-layer MLP. Regularization was applied with L2. There was dropout, and the optimizer was an Atom W with a 1E -3 to start with, and then with plateauing on reducing learning rate on plateau. And there's also early stopping implemented to prevent overfitting. So this model was applied, and when I saw the results, these were the overall observations that I noted. The first is that multimodal integration outperformed expression-only models. So I tried this first in an incremental fashion. So just tried expression only, and then added mutation data, and then gene essentiality data and lineage. And with each, there was incremental improvement in performance. And per drug R square shows that some drug classes are just inherently more predictable than others. Autoencoders provided more informative representations than PCA alone. I tried PCA alone, and autoencoders made a huge difference. So here's a key challenge when I was working through this. It is a small data set. So individually, there were, thousands of drug response data. There were thousands of cell lines that had characterizations in terms of gene expression. But finding the overlap of all of these brought the overall data set size down to 591 cell lines only, but there were 495 drugs. So how I tried to mitigate this was by feature selection. So using only mutations in genes known as cancer drivers, and then using only top 2,000 variable genes from the expression database, and dimensionality reduction to 64 dimension latent space for those. Heavy regularization, like point dropout of 0.6, early stopping, and 1E minus 2 for an L2 regularization. I do a threefold cross validation using 7 seeds and 5 model initiations per iteration. So, and at the end, what was also evident was that certain drugs are just not predictable. If their drug response was flat, for instance, there's no variability. So I filtered out those drugs that have low response variance. And here's the performance metrics on training and validation. This is something that was taught in class, and I was very excited to use it. So this represents average across 7 Cs, threefold cross validation, and 5 model instantiations. The training and test data for overall model loss looks much better than drug prediction loss. These are the other outputs that were also measured and optimized for expression reconstruction loss, which look better than the CRISPR essentiality reconstruction loss. And as far as the output goes, I saw a pretty comparable R squared in comparison with current published literature. Some drugs that are known to be predictable were also recapitulated in my data and my model. So as you can see that a majority of them are above 0.2, which doesn't sound that great. But given how noisy this data is, it's a good starting point. So performance on test data, what you see here is each box plot represents a drug pathway. And this is just sorted by the median of R squared across multiple folds. But what is notable here is that the wide variability of R squared across each drug pathway. So I tried to understand this variability and how much of it was technical versus biological. Turns out a lot of it is technical. So I told you I ran this across 7 seeds, and it turns out that some seeds just divide the data in a much worse way than other seeds. So if you look at seed 17, for instance, we get overall like high predictability. If you look at 42, it underperforms across all drug categories. And this is also, there's variability between different model instances and then between different folds of the same seed. There are other sources of potentially biological variability. So this is if you take just one seed and one fold and one model initiation, you can see that there is variability across different drugs that target more or less the same genes or similar genes. So the target here is BRD4, and here too the target is BRD4. They all belong to a certain category called chromatin remodelers. And you can see that they target similarly, but their predictability is extremely different, potentially due to differing mechanisms. And this one is extremely low. So there could be different mechanisms, different drug structures, different manufacturers, so on and so forth. This is just another example of how there's variability across drugs. Again, with this one fold, one model instance, and one seed, there are differing responses where the targets are the same. So you can see that the top ones are well performing. The bottom one, which is CDK1 and WEE1, CHEK1, these are all contributors to cell cycles, which means cell division. And as you can see, there's wide variability. So what are the limitations of this model? There is limited interpretability. So the first thing that someone would want to know, or that I would want to know, is what contributes to a particular drug's performance. Is it a high expression of a certain set of genes, or is it a particular mutation in a gene that is making something more or less responsive? So at this point, since autoencoders were used to reduce a dimension of these of gene expression and of CRISPR, it is hard to interpret down to a gene level. The other limitation is that with this particular model, I can predict all of the screened drugs, but if you introduce a new drug, it would be hard to predict without retraining the entire model. Gene or mutation level importance is very difficult to extract. I attempted gradient tape, but it just got extremely complex and are hard to interpret. So we can do an approximate attribution via latent feature like perturbation, etc. So we can knock down one latent feature at a time and then narrow it down that way. But that wasn't really satisfactory, with the goal being biomarker discovery. So here's what I would like to do to improve the solution. So I would first like to increase sample size by aggregating additional data sources, and then add self-attention for intergene interactions. I would like to explore per drug models. Now, each row is a cell line, so I would like each row to be a combination of a cell line and a drug. So this way I can incorporate into the input the structure of a drug, the chemical ID, or I can model response, or I'll be able to explain the response of targeted therapies, which a certain drug would only be effective against something with a particular mutation. I would like to learn and implement methods for model interpretability. I understand that this is a hot topic right now, and I think it's extremely important. I would like to evaluate this across multiple response matrices. Right now, it's on IC50, which is just one metric of drug response. There's others, there's root mean square error, there's area under the dose response curve, etc. I would like to build a model that is robust across multiple ways of measuring the response. With that, I would like to thank everybody for listening, and thank you so much to the instructors and the professors and teaching assistants for this deep learning course. I'm very grateful for the immense knowledge I gained. Thank you so much.